So, in today's video I will finally introduce you to synthetic biology, which is a very interesting buzzword that is going around today, and is a very nice approach to engineering biological systems. So, note that we are trying to systematically engineer biological systems. So, you want to predictably engineer biological systems. So, in today's lecture I will overview some of the concepts, what are the different synthetic biology parts that people have made. So, these are akin to the logic gates or circuits that electrical engineers make. So, can we make reliable parts? Can we make modules building on these parts? Can we combine modules together, and what are all the challenges involved in all of these, right. So, these are some of the concepts I will try and overview today. So, what is synthetic biology? A reasonable description, it is a bottom up synthesis of a biological system. How is it different from genetic engineering?
Way of changing an existing system.
Well, you still stick in new components and so on in genetic engineering as well.
They are not synthesizing it from its component parts.
In the sense?
In genetic engineering, you have a cell and you're adding something. It's more of like.
I can integrate a gene, no? I can take a new gene and stick it into a cell. That's in a way synthetic biology, in a way genetic engineering. The key idea about synthetic biology is that you try to be a lot more rational here, right? Meaning you expect that this is what's going to happen, and the key idea here is, can you make it more like engineering, right? So, in engineering you understand that if I stick in this module into this circuit, this is how it's going to work, in say electrical engineering or something. So, can you bring biology to that level of understanding, right? And people usually quote Feynman here, who said something like, what I cannot create, I cannot understand, right. So, another very key aspect is in some sense understanding those design principles we were just talking about. So, what are the design principles of biological systems, or how do you design biological systems that can have specific function, right, things like that. So, conventional genetic engineering typically focuses on tweaking one or two genes, right, or a few genes even. But synthetic biology is engineering driven and focuses on wholesale changes to the existing cellular architecture and construction of elaborate systems in a bottoms up manner. Right. And the idea is that new designs can be radically different, they do not need to mimic existing biology, they do not have to stay true to that, and they can potentially be designed to be more robust than systems fashioned by evolution, because anything fashioned by evolution, while undoubtedly very robust and so on, often has some artifacts because of its trajectory, right. There is some history associated with the system, right. And an important goal of synthetic biology is to uncover the design principles as we were just discussing, right. So, can we rationally design circuits that are very robust, that can exhibit a specific function and like oscillation or adaptation or a switch like behavior and so on in a reproducible, reliable fashion. And the important thing is that it has to work when you stick it into a system. So, this is always a challenging part. You build something in vitro, it works, right. What happens when you take it in vivo? When you implement it within a cell, does it work or not? So, the hallmark of modern technology is basically reliable engineering of many component systems, right. So, if you look at a system like this or a laptop or so on, there are like thousands of small components, but how they can be reliably composed to work together, right. In biology, this composition is the toughest part. You can't compose things. When you take A and B and put together, you never see A plus B. You will see either A square plus B square or A minus B something, not even linear, right. So, it is always going to be some really complex synergy or antagonism that happens when you put two things together, because there are so many ways in which they can interact. There are so many molecules, so many proteins and so on. And the synthetic biology challenge is, will the apparent complexity of living systems permit biological engineers to develop such electrical engineering like capabilities, right, where I know I take this module, take this oscillator, take this, I can make a pump that so you take all these components, you take this oscillator, take this module, put it, and maybe you can get a diabetes pump or something that secretes your insulin at specified intervals and so on. So, can you have this kind of a modular behavior wherein I know if I put module X and module Y together, I will get a particular behavior, right. So, this predictability becomes a very important aspect and is difficult to achieve. Right. So, let us look at what was the first wave of synthetic biology. So, the idea is very nice and lots of toys, like. So, what were the toys? People created small genetic devices like switches, cascades, pulse generators, time delayed circuits and so on, right. So, all these modules or all these parts become central to creating more complex systems. So, you stick all these. So, you want to build a more complex system, you need a a pulse generator or an oscillator or a switch and so on, and you stick them with in tandem with some other components, and finally you get an interesting behavior. So, what kind of strategies did people use? So, it was what they called iterative rational design. So, you construct circuit variants combinatorially and then filter out to those that have very good performance, right. You can do this using laboratory evolution and so on, right. You put a certain selection pressure, you eliminate those that do not show the desired behavior. There are directed evolution strategies, so on and so forth. So, this was one set of strategies that people used, and you may have heard about BioBricks and the iGEM competition and so on. So, what is a standard biological part? This is a nice definition. So, it is a genetically encoded object that performs a biological function and that has been engineered to meet specified design or performance requirements, and wait for the tough part, especially reliable physical and functional composition, right. It means that you design a system, you put these whatever proteins or whatever together, right, actually DNA, it is a stretch of DNA which will finally encode for some proteins whatever and so on, but if you stick this DNA into a system, it is expected to perform an oscillator function, right. So, it meets specific design requirements, and if you stick it into a system, it behaves as expected, right. And the idea of BioBricks was to catalog building bricks for biological systems or standard biological parts like all your electrical ICs and things like that, using which a synthetic biologist can program living organisms, right. So, you put an oscillator, put this, then it is going to behave in a particular fashion. So, the BioBricks Foundation hosts lot of interchangeable parts and so on. So, then there were a lot of modules that were built. So, there were some transcriptionally based modules. For example, this module is dependent on arabinose and so on. So, if arabinose is present, it will behave in a particular way, there will be a GFP that is triggered, and there is some, it also interacts with the LACI gene and so on and so forth, right. And they found some oscillation in fluorescence. So, this particular transcriptionally controlled system exhibits oscillations as you see in the bottom, right. So, there are people who have built a relaxation oscillator with fine tuned oscillatory dynamics, and it is not sufficient if you build oscillators, are they fast and are they tunable. Okay, by tuning we mean that you can change the amplitude or frequency especially, right. Can you change the frequency of the oscillations or even the amplitude, you want to tune the circuit to respond in a particular fashion, right. There are translational modules where there are ribozymes or riboswitches. So, ribozymes are basically RNA molecules that can act as enzymes. So, they can RNA can fold into nice loops and so on, and there are regions of RNA there are regions of RNA that can actually bind certain ligands. So, you see there is a ligand that there is a theobromine binding domain in one of these RNA and so on, and they also built circuits that they could act as a NAND gate, NOR gate and AND gate and so on, right. Only if both RNA bind, then a particular response is produced, or if both bind, so an AND gate kind behavior and a NOR gate type of behavior and so on. So, you can see what is the output, right. If both are not present, there is no output, if both are present you get some output. Even in these you need to get 0 output, but this is obviously not a digital gate, it is a little analog. So, these are low values which are essentially the equivalent of off, and this is a high value the equivalent of on, right. So, and this uses various biology concepts such as siRNA and RNA aptamers, and you can also integrate multiple signals, right. So, the NAND gate integrates 2 signals and then knots them, NOR gate adds 2 signals and then knots them, AND gate just multiplies 2 signals in some sense. And there are also post translational modules. So, if you go back to the very beginning of the course where we discussed the flow of information, that is transcription, translation followed by PTMs or post translational modifications like phosphorylation and so on, and things like phosphorylation give you very good fast switching behavior, right. You can quickly switch something on, switch something off and so on, and so they are very useful. So, if you see there is negative feedback which works in a particular way, there is positive feedback which gives you an amplified response and so on. So, they designed very nice receptors that like this which can give you interesting behavior. So, they had an orthogonal ribosome RNA pair, mRNA pair that could encode for even synthetic amino acids, right, not just the 20 amino acids. There are even very interesting studies that have been done which use a different translation machinery. So, no longer 3 base codons, but 4 base codons, right, which have a which can potentially be an orthogonal translation machinery in an organism, right. And another very interesting concept is that of synthetic protein scaffolds, right. So, this is especially useful in say polymerization reactions and so on, or in any pathway wherein you have, let us say you have A, E1 makes it B, E2 makes it C, right. So, the idea here is you take a DNA and you use it as a scaffold and you put some nice protein molecules on it. So, this could be your E1 and this could be your E2. So, this will facilitate better reactions, right. So, now A goes in, comes out as B, goes in spatial collocation, comes out as C. So, these enzymes are stuck onto a DNA scaffold to bring them closer and to improve reactivity and so on. So, there are very interesting studies which have used concepts such as these, and they have also studied synthetic signaling pathways with different kinds of interactions. If you see the the signal binding domains in proteins are very modular, and so there are people who have actually swapped, mix and match those domains to create artificial signaling pathways which work very differently and so on. But basically there are modules that are built on transcription, translation as well as post translational modifications. I am just giving you an overview here, you can always go back and read a lot more in some nice papers such as these, right. The next wave of synthetic biology is combining these modules. How do you put these modules together to build more complex systems, more complex functions, and this is a nice slide telling you the milestones of synthetic biology. So, the first bacterial toggle switch, and then you know the IGEM competition. The first IGEM competition was in 2004, and there is a nice strategy wherein bacteria were designed to, this was called a pathogen find and destroy, right. It could search for pathogens and then destroy cancer cells. This was cancer cells, so they could sense a hypoxic environment. There are also studies which try to detect a pathogenic organism and then secrete certain, because in things like cancer or even in infection, the difficulty is in selective destruction of the disease cells, right. So, how do you do that? So, in this case they try to selectively detect cancer cells by searching for hypoxic environments, because in cancer there is a lot of hypoxia, right. And then there is artemisinin, we will try to study that. This is a little old, it still talks about the 5th IGEM. We regularly have teams from all over the country, even India, participating in IGEM. So, last year I think IIT won some very good medals, this year IAC won the gold medal and so on. These are the local chapter gold medals, and there is also the final contest in Boston which is far more competitive. And yeah. So, now we are slowly moving towards combining basic parts and modules to create systems level circuitry. But the problem here is, this is something I probably showed you in the very first lecture of the course, more is different. So, this was Anderson in 1972, a Nobel laureate who saw things way before others did. So, he said psychology is not applied biology, nor is biology just applied chemistry, right. See, biology is nothing but a combination of various biochemicals, but there are so many emergent properties that occur, and the whole becomes not merely more, but very different from the sum of the parts. And this is the challenge that one needs to tackle when you look at even synthetic biology, or of course systems biology itself, right. You have a very complex system, it behaves very differently from the basic modules that make it up, right. So, beyond the regular standard design issues, you must worry about things like cell death, crosstalk between different modules, right. There are some molecules, some chemicals, some signaling molecules present in different parts of the cell, and they could all be interacting with your module and other modules. Mutations that can happen spontaneously, the cell might just try to kick out your module in the first place, right. Inter and extracellular conditions, noise which becomes a major challenge, right. There is so much of heterogeneity in cellular gene expression or in any of the other components within a cell and so on, and various other biological phenomena, not in the least evolution, right. The cell can just mutate, change, evolve, behave very differently after a few generations, right. And complexity increases really non-linearly with the number of components, right. So, there is also very interesting studies that have come up which talk about synthetic multicellular systems. Some of them are still toys, like for example, there was this system wherein an adenine utilizing enzyme was destroyed in one, and a lysine gene was removed in the other, and these two, so this cannot produce adenine, this cannot produce lysine, and they were made to obligately cooperate with one another, right. They are obligated to cooperate because they can't survive otherwise, right. So, this is a very interesting study. And then there are many quorum sensing studies which leverage noise in the levels of various proteins to maintain a stable population and so on. And very interestingly, they also tried some inter-kingdom interactions, right. So, there is E. coli and yeast. So, not just, so this was just 2 yeast cells, this is just all bacterial cells, but this was E. coli and yeast cells which try to find a particular kind of cell and they induce parasitism and so on. Like, can you then destroy a particular cell and things like that. So, many interesting studies, a lot of them based on quorum sensing, because if any cellular communication involves a lot of quorum sensing and so on. Right. And then there are some nice therapeutic tools that have been built, so which for example destroy tumors by detecting a hypoxic environment in the first place, and there are bacteria that thrive in tumors using quorum sensing to detect high population density, and then they express invasin which bind mammalian receptors and initiating cell destruction in endocytosis. Right. So, there are some very interesting applications of synthetic biology. Another classic metabolic engineering application is how they stuck this pathway from artemisinin, from Artemisia annua, a particular tree, to produce artemisinic acid. So, if you see they produced artemisinic acid through metabolic engineering followed by semi-synthesis. This is chemical synthesis. So, they could not make artemisinin directly, but they found a way to actually make artemisinic acid by integrating these genes from Artemisia annua into Saccharomyces cerevisiae, followed by chemical synthesis of artemisinin, and this is a very important anti-malarial drug. But I have recently heard that this has not been a very stable system, right. Although this engineering was successful in the beginning, but there are some issues when you try to scale it and so on. So, there are always challenges that we need to worry about. And the other very interesting aspect is how do you design minimal systems or minimal genomes, right. This is one interesting direction for synthetic biology, because if you want to engineer an E. coli or a Saccharomyces cerevisiae cell, you already have to live with all its existing complexity. And an E. coli cell already has 4000 genes, and into which you are integrating your one new module, and it has to integrate with interact with all those 4000 genes and still survive and so on, right. So, could you build a minimal genome, right? Could you build a minimal genome with say 300 genes or 400 genes? There is still a large number, but potentially less interaction, less complexity to worry about, right. And there are two interesting sets of studies that have been done. So, one is top down elimination, wherein there are groups which have been knocking out genes from different organisms, and then there is also Craig Venter, who has been I think some of this work is already published now, which talks about say Mycoplasma laboratorium based on Mycoplasma genitalium. Mycoplasma genitalium is already a very fastidious organism. It is a very small genome, and it's pathogenic, and it can only grow in certain conditions and so on, right, which means that it needs a lot of nutrients to survive and so on, because it can't make too much on its own. And they made Mycoplasma genitalium encode the genome for a new organism and start producing this organism. It is a very interesting set of studies, and there are two very nice books if you want to read about synthetic biology. There is Regenesis, and that is by George Church, and there is I think Life at the Speed of Light by Craig Venter himself. So, Craig Venter is the genome sequence expert. So, and there are also groups that are trying to do bottom up forward engineering. So, how do you put up a minimal cell wall, how do you just integrate some basic protein synthesis machinery, or can you have synthetic vesicles and so on. And there is also the other, synthetic biology is becoming an ocean now. So, there are courses on synthetic biology, I can't do much justice to synthetic biology in a lecture, right. But the other interesting concept is that of cell free synthetic biology. So, can you have essentially in vitro synthetic biology, you do not have the full cell, but you have a circuit that just operates within a test tube literally.
Minimal genome, so so we are eliminating some of the genes, I think. So the redundant
robustness will go away, but yeah, so robustness will go away. In fact, they also found that the ATP maintenance energy also went up and so on. So the organism becomes much more fastidious, much more fragile, and it needs more pampering to survive literally. And there are many interesting research problems and challenges, as you would expect in a nascent and young field. So, the major challenges that limit engineering of biology arise from A, complexity, very difficult to handle, and still the characterization of the systems is insufficient, it has even synthetic systems, and there is so much of inherent heterogeneity, right. So, if you try to stick a system in or even look at a single gene, in a population that gene will have a curve, a distribution of expression, not a just nice spike, right. So, because of these complexities it becomes very challenging to model such systems, and of course evolution itself, right. The system will just evolve to a different state or reject the changes that you are putting in and so on, right. So, importantly, models which consider system level effects and crosstalk between parts will aid in building better devices. So, in fact, this is something that some of the folks in my lab are working on, like how can you consider what is known as retroactivity, right. So, you have a system, you integrate it into another system, there is retroactivity, right. The fact that you have integrated into the system has a pull on the other system, which basically causes a different behavior, right, not the desired behavior. You have an oscillation initially, but you stick it in the system, the oscillation dies down. But there are some systems that seem to be inherently resistant to this kind of retroactivity and so on, right. So, how do you standardize? The goal is this, right. In fact, if you see any of the synthetic biology columns or magazines and popular science articles, the idea is they will say that you just sit on a computer, you key in some genome sequence, you then order it and you get an organism that can do something cool, right. So, we are far from that, but that is the eventual thing that we want to work towards. Right. How do you reliably compose or combine these standard biological parts? So, the BioBricks and so on, and very importantly what kind of context can it work in, right. So, you build an oscillator, you stick it into one cell, it works beautifully, you stick it into another cell, it does not work at all, right. So, depending upon the context the behavior might vary, right. Then noise is another major factor, right, and noise arises from several sources. It could be environmental variations, generic fluctuation in gene expression, what stage of the cell cycle the cell is in, maybe there are some metabolite concentrations which are massively influencing it, and mutation, evolution and so on. And one can counter noise. So, there are systems, there are places where noise is an advantage, right. For bacteria that are resistant, noise is a great advantage, right. So, you have 100 bacteria each expressing different levels of a particular protein protecting it from antibiotics. Those which express a higher level of them will survive when you throw a drug, right. If everything express the protein at the same level without noise, then all of them would have died, right. But the ability to fluctuate their gene expression levels gives them the power to survive, right. So, and there are interesting studies where noise has been leveraged for population control as well, and there are some regulatory networks that are very robust to noise, right, and there is a lot that we can learn from these kinds of complex networks like circadian rhythms. And then there are epigenetics. Maybe we should not go into that too much because we have not done a lot of biology in this course, but there are many heritable changes that pass on from generation that propagate without changing the underlying DNA, right. So, maybe there is a methylation of DNA which passes on, right. So, how do you can you circumvent or exploit these epigenetic processes, and can you design cells that will modulate the epigenetic memory and so on, can you just remove it or preserve it, right. And computational analysis, this is why we are talking about synthetic biology in this course, right. A lot of background work for synthetic biology comes from computational and systems biology, right. How do you model these systems? So, like ensemble modeling or like sampling the parameter space and topology space to identify reliable circuits and so on. So, we can still construct reasonably good models of single circuits. You can also analyze their robustness and you can make reasonably good predictions, but how do you handle very large systems, right. If you wanted to make a 7 node oscillator, there is no way you can exhaustively sample. So, if you are talking about a 7 node oscillator, you are talking about something like 3 to the 49 circuits or something like that, right, and that is some astronomical number, right, and you can never sample that space. So, how do you go about constructing such circuits? Many interesting challenges, right, and how do we model crosstalk between signaling, regulatory and metabolic pathways? This becomes another major challenge. So, we will talk about some of these when we wrap up the course. Can we build modular plug and play models? This is like the final aim of synthetic biology, right. You want to just have modules, have BioBricks, have DNA sequences, cut paste, it all works beautifully, right. There are some very ambitious ideas such as genetic engineering of living cells and executable biology, right, wherein you basically use a high level engineering language like your all the electrical engineers use, right. There are many interesting design languages that electrical engineers use to design systems and so on, right. Can you do something like that? And safety remains a big question mark, right. So, there are concerns, valid concerns, less paranoia, but I think scientists are still working actively to resolve these. There are many challenges, but there are many advances as well. So, I will not go into these, but there are very interesting things that people have been doing in the areas of safety. Lastly, some open problems. You have an in vitro system, you have an RM, you have an in silico system. What happens when you stick it in vivo? Can you handle retroactivity, right? Or can you design adaptable or evolvable systems, right? Or so, see classic systems level engineering practices, can you apply them all in biology like modularity, component testing, standards, interfaces, parts library, computer aided design, right? You want to apply all of them in biology, but it is very challenging because biological systems are unique. Why unique? Highly nonlinear, lots of noise, lots of robustness, evolution and heterogeneity and so on. And of course, more is different, right? You add two things together, you get something completely different. And but one needs to explore new computational methods to try and design robust systems in the first place, right? If you design a robust oscillator that is also non-retroactive, it is going to likely be a very good synthetic biology module that you can reliably stick into a complex system, right? And as Tom Knight said, the goal is to finally tame biology as an engineering discipline, right. So, biology is still not a truly an engineering discipline because there is so much, how much of it is art and how much of it is science, right. So, any genetic engineering or synthetic biology experiment becomes quite an art rather than a systematic, you integrate this with this, it just works, right. So, that is where it would become a science, but that is where all the excitement is. Okay. And hopefully that is where we will see the third wave of synthetic biology, wherein we see mostly some interesting success stories which combine all these modules, complex cell communications, complex circuitry to solve real life engineering problems. So, this was like a quick introduction to synthetic biology. So, in this very brief introduction to synthetic biology, I hope you have had a feel for what are the kind of parts that people have built so far, and how some modules have also been built, and how one can try and combine these modules to build circuits that perform desirably. So, the dream is to have a computer aided design of biological systems, wherein you decide that I want a functionality X, and I figure out what is the DNA corresponding to that functionality X, and generate the DNA sequence, stick it into an organism, and it works perfectly. Although we are still a long way away from that, we have been making very interesting progress towards that, and the very interesting papers that I showed you today discuss many of these interesting studies. So, from the next video onwards, we will look at some advanced topics, beginning with this very interesting concept called ensemble modeling.